Welcome to Pediatric Grand Rounds. Thank you for joining us today. We are in the Mill Mountain Conference Room at Tanglewood Complex. Please join us if you're nearby, and thank you for joining us online. Today's speaker is Dr. Brittany Howell. She's an associate professor at Virginia Tech, where she leads the Maternal Influences on Neurodevelopmental Lab Mind at the Fralin Biomedical Research Institute. The research blends biological and behavioral science to understand how maternal health, behavior, and environment shapes the infant brain and behavioral development. Dr. Howell's work is dedicated to improving maternal-infant health outcomes by exploring factors such as nutrition, stress, sleep, and early caregiving with the goal of supporting families and optimizing neurodevelopmental outcomes in our communities. Please join me in welcome Dr. Brittany Howell.
Good morning, everyone here and elsewhere. We have semi-limited time, so I want to make sure we jump in. It's always weird to hear people talk about you when you're standing next to them. It makes me uncomfortable. I'll get used to it maybe someday. All right, I start every talk with thanks to the people who actually make it happen. So my research group has kind of grown majorly in the past couple of years. Clearly, I'm not going to go through everyone's names, but I can't start without making it very clear that everything that I'm going to show you, whether it's our data or others' data, comes from a research community. And so I just always have to start with gratitude to them and all of the effort that they put into it. And I'm going to talk about what we do as far as MRI specifically, and you'll get a real clear view of why I start with thanking them, because it's somewhat thankless work. All right, I have no disclosures, no conflicts of interest of any kind. So I want to ground this in, especially, so I know this is, a clinical grand rounds, so I'm going to try to keep it clinical, but, I'm a researcher. But I do think that we as academics and you as, physicians, clinicians, and academics potentially, right, have a shared goal. And that really is to help, you're helping to use the empirical evidence that we are producing, hopefully, right? That's the idea. And so my lab is really trying to provide that empirical evidence so that we can help families just make choices. So I'm not here to tell families what to do. That's what you guys get to do. You get to talk through risk and benefit and help them make those choices. I'm here to help guide those choices with really robust information. And at the end of the day, we really all just want all kids to be happy and healthy. So we have that common goal in mind. I also, you'll see throughout, have gratuitous pictures of my own children. They're not as cute as they used to be. They're 7, almost 8, and 13 now, so, But I like to be reminded that they were cute at one point. So as Dr. Parmeshwar said, I am a researcher, and these are the questions that kind of guide my lab. And so I'm not going to go into them specifically, but what I will say is that we're going to touch primarily on the mother-infant dyadic interaction piece. And again, I'm going to try to keep it relevant for clinical things. But the one that we're going to focus on is the efforts that my lab is putting into making the neuroimaging specifically. I'm going to focus on infants because that's the space that we live in, but that trying to make it accessible to all populations. So there are lots of reasons why current neuroimaging methodologies won't answer the questions that we want or need to answer, and they're just not accessible to everybody that needs them. And we'll talk a little bit about why that is. But that's one of the main foci of my group. Right. So why study brain development? Hopefully I don't have to convince you all of this, but in case there are some non-believers, I don't know. I wanted to start with this idea of developmental programming. So it's just this idea that parts of your body that are changing rapidly are going to be sensitive to external influence, right? And the brain is a really great example of that, not just because it's sensitive to, but that's kind of its role, is to adapt to the environment and to learn, right? Change based on the kinds of exposures that it experiences. And so this is really a feature, not a bug. And I think oftentimes it's considered a bug, right? So that's vulnerable. It really is sensitivity, because that's what the brain needs to do. It needs to figure out what is the environment that I'm in, how do I need to grow to meet the demands of that environment, and that way I will be successful, right? I also wanted to highlight, again, why I focus on infancy, and that's primarily because there's so much growth that happens postnatally, not to say that there isn't a lot that happens prenatally, but when you're looking at rates of growth for both subcortical and cortical gray matter, it peaks at 6 months, which is As a side note, when babies are meant to have only exclusively been exposed to breast milk, I'm not going to talk about the nutrition arm of what I do, but I like to plant that seed because when we're thinking about brain development and how much happens during that time, it's pretty remarkable that infants or mammals in general have evolved to be supported by a fluid that is produced by their mothers. And there's something there that we really don't understand, at least as scientists. If clinicians have some insight, please share with us because we feel like we're fumbling around the dark. And these are just white matter volumes. And then, so white matter volumes, the plot in the middle, that rate of growth peaks at about 2.5. Not a huge, that age doesn't really matter, but again, the point here is that early development of the brain happens very, very quickly. And so this is a time where if we don't understand what's happening, we could have implications for the entire lifespan that we aren't able to really understand or mitigate later. So that's why we study it. Clinically, again, you know more about this than I do, so I'm not going to belabor this slide in any way, but just to point out that, of course, if you want to think of it more directly, why do we care? It's because there's a lot of injury that can happen surrounding birth. And what we don't really understand is how do we identify what kids early on are going to need extra support, right? So I'm a neuroimager. How can we use neuroimaging technologies to identify which children experienced these kinds of insults? What do they need to have optimal development, right? And part of the issue there is we don't have great methods, right? So we don't have methods that we can capture characteristics of the brain that we think are important for those long-term outcomes. Also, the whole thing with funding only lasting for 5 years in an NIH grant and kids taking longer to grow than 5 years, right? Depending on your outcomes. That's also a barrier here. But the idea is that we really need to understand what's happening in the brain, whether it's, again, perinatal brain injury or just kind of how is the system working when it works. And that way we can identify when we really need to be paying attention when you differ from that normative trajectory. And that's still ongoing active research. All right, so I'm going to mention ultrasound only once, and that's only because I don't do ultrasound, but it is, of course, the most common neuroimaging method used in pediatrics, especially infants, right? And so these are some data that a colleague of mine, she did this during her dissertation work at Oxford, I believe, and now she's working with some friends in Copenhagen. But sometimes I don't think that we can appreciate what information we can really pull from ultrasound, and you can get pretty detailed images from ultrasound. So, when we're doing regular, you guys are doing regular 20-week pregnancy checks, you can go even deeper than that. We can look at things like volume and sulci and gyri using ultrasound, which is pretty exciting. And the one thing I will point out about these data is these are not research data. They pulled from clinical data. So you can, with the images that are being collected now, you have to adjust kind of how those data are saved so that you can look at things in the detail here, but it is possible with that technology. You will notice, though, that it's not particularly detailed, and of course, right here, we're just looking at a surface reconstruction, but even if you look at the top rows where you're seeing inside the brain, the contrast isn't great, right? You aren't able to see structures particularly well, and that's where other modalities, specifically MRI, can start to fill in those gaps, which is what we'll focus on today. So the two modalities that my lab uses specifically are non-sedated sleep MRI and then wearable magnetoencephalography, which we'll talk about towards the end. But I want to start with MRI because I think it's the most relevant and probably the most, again, the only way that we can look at anatomy without exposing kids to ionizing radiation. Right. So sometimes CTs are required. Absolutely. You all are the ones that work together to make sure, you use whatever modality is most accessible. But research-wise, I would never ask a parent to expose their child to a CT just for funsies, right? And we don't have to. We can use MRI. So this is one example of one individual scanned at 3, 9, 12, and 24 months during non-sedated natural sleep. And I will say that we were supposed to get a 6-month scan and she didn't sleep. And I've just been lazy and haven't updated the slide because we have other kids who did sleep for all 5 time points. But her data were so beautiful and just showed that, make the point, again, kind of going back to talking about that programming part of how drastically the brain changes between 3 and 9 months. And there are some practical considerations here. So if you look at the very first image on the top left, So that's our 3-month T1. How often do you all look at MRIs? Often? Ever? So when I say T1, does that kind of make some kind of sense? Perfect. And of course, don't worry about the physics. I don't know the physics, full disclosure. I know sequences, but anyway. But the idea here is that with a T1 and a T2 structural image, you can actually see the underlying anatomy. And that's interesting because we can actually see how the brain is changing, right? We can see how its shape is changing, how the volumes are changing. Then we can relate those things to behaviors. However, when we start to think about how do you process these images, it gets kind of tricky. And that's particularly because very early on, I don't know if you noticed how much darker the 3-month image is. And that's not because the actual tissue is darker or anything. It's just because there isn't as much signal. And so we can't make up signal. There isn't a technology that can just amplify that necessarily without potentially adding some bias. And so what we have as researchers have to do is figure out how do we process these things so that we can get the information that we need out. Once they get to about 9 or 12 months, the T1s are great and you can get all the information that you need. Typically, T2s are good for white matter lesions and things like that. So if you've ever gotten a report, typically you'll have a T2 as well. Ours are a little longer and a little higher resolution because we're asking questions about very specific ways the brain is changing, and so we need higher resolution data. Then the bottom row, that is a fractional anisotropy image, so that's something that we can get from diffusion MRI. The beautiful rainbow spaghetti picture up here, which is really hard to see on the TV. But the idea here is that not only can we say what is white matter, like we can in the T1 and T2, but we can actually look at the orientation and the directionality. It doesn't tell us anything about communication between the two different regions. It just says that they are structurally connected. And that's important because especially, and we'll talk a little bit about this in just a moment, white matter seems to be particularly sensitive to all sorts of insults. And that's one of the reasons we think that because it's changing during that time, during early life. All right. Okay, so what are some of the challenges in getting good MRI images? And the main one is movement. So I'm not going to belabor the one on the left, so it's kind of hard to see on the screen, but hopefully you can tell that the image C is really awful, right? You can't see anything. And I promise their brain doesn't look like that, right? And that's because when you're imaging anything, even adults, if they move within the MRI, it smears the image. And that means we can't see anything. So that's a huge issue for pediatrics because they move constantly. And most of the time, you can't ask them to hold still. We've had studies where, so I've scanned from 0 to 5 years, and the older kids will do their best to hold still. And you'll ask them to hold still, and they'll sit there for about 15 seconds, even the really good ones, and then they'll ask you if they're done. And this is kind of frustrating when you have even if it's a 4-minute long sequence where they have to hold still. And we can play movies, but you have to find ones that don't make them want to sing along or move their mouths. So all Disney is off the table because all of those have songs or they want to dance. So it's really challenging to get good awake data, right? So then what do? We sedate them, right? Which again, I'm not saying that sedation is unnecessary in every case. That's up to you all as you care for your patients. But this is for this plot is specifically looking at, I think it's what I put my notes, about 340 centers from across the world that scan neonates. So this is within the first couple weeks of birth. And so you all dealt with neonates. They eat, you clean them, they sleep. Right? And they don't have a lot of musculature, right? They're not going to be moving their heads, they're not going to be rolling, they're neonates. So my point is that these are some of the easiest cases as far as motion goes, right? You can pad them in, they don't move all that much. And still, in even this situation where it's pretty, the most ideal situation we have for motion in the US, you still have centers that are using sedation 25 to 75% of the time. And of course, there are other parts of the world where they're always sedating their neonates. And you might think, well, does that really matter? Because of course, again, we need to get really good, crisp images, right? And it does matter. And so this is one example of work that I did looking at what sedation does. This is in monkeys. So we wanted to, of course, control for everything that we could. So the panel in the middle. So, we work on the TV. Here, Here we go. So this panel here in the center, these are animals that were exposed. Well, the image that you're seeing was the 5th time that they were sedated for scans, because monkeys, although there are some adult animals that have been trained to hold still in a scanner, infants you can't train them fast enough in order to capture the brain growth that we're interested in, so we have to sedate them. And so the middle panel, those are kids that had been sedated 4 times. Again, this is the 5th time, the image that you're looking at. And then that bottom panel, these are age-matched, sex-matched controls that had never been sedated before. And so the question we wanted to ask was, does that repeated exposure, as part of research, we were trying to see how their brains were changing across time. That specific study was in the context of maltreatment. So we were trying to see, is it something about the experience of maltreatment that changes the brain? And what we found was that in addition to what we were assuming, and of course we had controls, and so we were looking within the same kinds of exposure. But the question was, is that exposure itself doing something? And if it's doing a lot, then it's potentially masking any of the interesting things we're trying to uncover about the experience. And so the naive animals have overall higher fractional anisotropy. So if you remember the image before, all that metric is saying is kind of the coherence of those white matter bundles. And so that's impacted by things like number of axons, the level of myelination. It's not a proxy for myelination, but it does give you an indication of the kind of the structural integrity, I think, is the kind of buzzword that people use for fractional anisotropy. And what you see in those kids that were exposed multiple times, I'm sorry, I call baby monkeys kids, it's a terrible habit, but they are, so the purple in these bottom plots, those are exposed. So again, they had been exposed 4 times, and this is a set level of exposure as well. So again, this is very controlled. That was the idea. That's one of the benefits of using animal models. And you see reductions in fractional anisotropy in the genu of the corpus callosum. So that's the very front part of the corpus callosum being the largest white matter tract that connects the hemispheres. And it's one of the places that's growing very quickly, actually during early postnatal development as well. And then there's just another example of the left optical tract. So point being that we know that sedation impacts the brain itself. And so if we can avoid that, why wouldn't we, right? Again, I'm not trying to say that you should never sedate, because that's up to you all. You all know better when that is clinically indicated, but I think it's important to know the risks and benefits in as detailed a way as possible, so that you can make those kinds of calls. Okay, so we know that sedation isn't the ultimate solution, right? There are some things to be considered and balanced there. So what can we do? So on the technical side of things, we're trying to figure out how do we correct for that motion. And fortunately, there's some really, really smart computer scientists who are working really hard to be able to deal with this kind of motion in real time. And so that means that we could have, hopefully, an awake, behaving, not like moving, but if they're just being asked to hold still, or if they want to move their mouths while they're watching a Disney movie, we can handle that. And that way we don't have to sedate them. We just need them to lay in the scanner. That's still a challenge. I don't know if any of you have had MRIs. It's not particularly comfortable. Babies don't care because, of course, we scan them to sleep and they're used to being kind of cuddled in dark spaces. So they're pretty easy. But anyway, again, if you can appreciate the difference from C to D, D is the corrected image. And so they were able to take this really terrible, can't see hardly anything image, apply some advanced computing, and get us to a place where we can actually see some anatomy. And that, again, is really, really remarkable and is really important, I think, clinically, as we try to move away from just defaulting to sedation. Okay. So I'm going to shift gears just a little bit to talk about non-sedated sleep MRI. There are some pediatric imaging centers that are trying to move away from sedating older, I say kids, but when I say older, I mean like 3 months and up, so beyond neonates, right? So the kids that are going to be wiggly and that you can't ask to hold still, they just clearly won't understand. And so how do you do that? And the best way we have now, until these motion corrections are implemented by the major scanner manufacturers, because that's a whole other issue, is getting them licensed by the manufacturers, making sure that the scanners that they're using can implement these algorithms. All the business stuff is a big headache. I'm sure you all are very used to dealing with big business and things like that, so this is no different. So in the meantime, at least for research, what we try to do is do non-sedated sleep MRI. And I'm going to blow through this because I don't know that it's kind of the meat, but it's something that I'm really proud of because we work really hard to get these images. And so the first thing we do is just make our families comfortable. What you can't see is there's a changing pad, a TV. What else do we have in there? We have an MRI safe crib, but the idea is that it feels like a living room. We just want them to come in and be comfortable. We are, I'm sorry, but trying to separate ourselves from hospitals. We're trying to say we're not a clinical space. Because that, depending on their experiences, that can bring up some stuff, and we don't want to muddy that water. The other thing is we work with a lot of specific clinical populations, and we want to make sure that in their minds it's completely separate from their clinical care, and make sure that they, even if they think that we are somehow involved, we are reinforcing that we are absolutely not. Their care is priority, even for research, and so we want to make sure that we're separate things. Another thing is that we'll be there for hours because we scan them during natural sleep, and typically we'll do that in the evenings because that's when they sleep deepest and longest. And we need, at least for our research protocols, about an hour, ideally, of good deep sleep. Here's an example. This actually isn't our setup over at FBRI. I haven't taken pictures. I probably should, but it looks pretty much the same. And the only thing I wanted to highlight is that we had to make a lot of adaptations, so we're covering all of the lights. We are using all plastic everything. That's just a standard changing pad. This guy is a pillow filled with chickpeas. So when you have tummy sleepers especially, they need that kind of pressure. And using a weighted blanket doesn't give them the same kind of pressure. So the chickpeas, we can move them around and put them on their sides, or we can kind of prop them up a little bit. The idea is we want them to feel as comfortable as possible so that they stay asleep. It's kind of a guessing game. And the other thing that I'll point out are these headphones over here. So the acoustics are the biggest risk in MRI. So it's not ionizing radiation. There is radiation, right? So we are depositing energy into whatever we're imaging. That's true for MRI in general. The issue there is potential heating. That part has been figured out. People have looked at what does the temperature look like inside an infant's head, inside an adult's head, inside an adult, whatever body part you're trying to image. All of that kind of safety stuff has been done, and it raises the temperature by at most 1 degree centigrade. That's been across the board, all of the studies that have been done. And so that's considered safe. Again, you have to balance all of the things. The other thing that the scanner does is it actually won't allow you to scan them long enough to increase their temperature that much. So there's a specific absorption rate safety measure that's in there that if you are trying to run a sequence that will heat them more than is safe, it just won't run. And so there are lots and lots of safety guards in for that piece. The part that is not inherently kind of protected by the scanner or anything inside the scanner itself is sound. And so the scanner can get up to about 90 or 100 decibels. So 60 is about a normal conversation. That's about what the sound I'm making right now. And that's what we want to bring them down to. And so to do that, we have at least 2 layers of hearing protection. So we actually used Swimmer's silicone earplugs, and that if you get a nice good seal, drops it by about 20, 22 decibels. So that gets you down to, again, if you're starting at 100, that gets us down to about 80. And then we have active noise canceling headphones. You can't really see them on the side here, but these are headphones that will actually sample from the acoustics of the scanner, and then we'll actively counteract them. So then we can bring it down further to about, I think it sets at about 72 if we have a nice good seal. And then again, you subtract 20 from that because it's a linear relationship, and you get down to about 55. So really, they're being exposed to what they would normally be exposed to if somebody is holding them while they're sleeping. That's the goal. Part of that is because we want to make sure that they're safe, and there's some evidence that neonates won't show that when they're exposed to loud sounds, and so we don't want to hurt their hearing and not have them tell us. Older kids will tell you immediately. We can tell when headphones are not on. Well, they're not shy. There's still some room for innovation though. So, we were just talking about neonates and some of the little ones. When you start to get to toddlers and kids that don't sleep in arms and are used to being put down and going to sleep on their own, we had to come up with some bespoke tools. So the picture on the far right is the initial crib I built. It's just made out of PVC pipe and plastic chicken wire. And the idea was we wanted to give them a safe way to sleep on the bed. Of course, we had to take it off before we put them in, but the goal was to not have to transition them. Because I don't know if you all have had to try to move children from car seats or beds or whatever. If you have to lift them up and then set them down, it's that vertical movement that seems to trigger something with your vestibular systems, and then they wake up, they startle. And so if we can avoid that, then we're much more likely to be able to get them in the scanner and get some good data. And so the middle is the actual schematic, and we actually had it built instead of me making it in my living room. And it's much lighter. It's made out of carbon fiber, which feels really sciencey and fun. And all it does, it's super simple. It just clips into the bed, the scanner bed. You don't have to do anything different to the scanner bed at all. And then it wraps around the bottom with nylon webbing and clips. And so that's the picture on the left of the finished one. People can buy these now. I think the price continues to go up. So I think it's about like $8,000 right now. But again, if it prevents us, at least for us, for research, it increases our success rate. And again, if this is something where, as clinicians, people want to move towards awake scanning, this is a tool that can be really helpful. However, it does not solve all your problems because scanning still takes forever. It can, right? So our protocols are typically designed to get everything we need in about a half hour, but we always plan for an hour. So because we have a sleeping baby, we will continue to collect data up to an hour, because why not, right? We've spent a lot of effort, families have spent a lot of effort getting them to sleep, and let's optimize let's leverage that as much as we can. So these are just a bunch of different scenarios of kind of what a non-sedated sleep scan might look like. So case B is kind of the ideal one, right? Family comes in, you do all their safety screening. If we need to get height and weight on baby to put into the scanner, so that's for that SAR level calculation. Then they'll be awake for a little bit, sleep for a little bit. We get them in the scanner, we get an hour of data, and ta-da, we're done. That happens maybe 5% of the time. The other times, it's one of these other flavors. And the only point I want to make here is that this is one of the main issues with transitioning any of this non-sedated methodology to the clinic, right? Because the clinical scanners don't have time for people to spend 2 hours getting one kiddo to go to sleep. And I totally acknowledge that. And that's why I'm really excited to see that we're working on the other side of things to get to be able to collect data that we can then correct for any of the motion or any of the other artifacts. Because I don't think that this will ever be something that, unless you have a lot of resources and you can, dedicate it. But it's hard. But right now it's the best we've got. All right. So I want to talk a little bit about incidental findings. So this is somewhere where research and clinical care very much directly interact, right? So for every scan that we collect, we have a neuroradiologist read them. That's baseline. I think it's ethical, especially given that we could identify something that wouldn't have otherwise been identified that we can address very early, or at least make a family aware and their providers aware so they can track that child in more targeted ways, right? So this is one example. This is out of the Developing Human Connectome project, which is the a European project that has scanned thousands of babies now starting right at birth. And so these are all neonates. This was 500 of their asymptomatic term neonates, so no preterm, no behavioral issues, no indication of anything else. And so when you're looking at the rates of incidental findings, in about 47% of the babies that they scanned, they found something that they identified as an incidental finding, right? And so an incidental finding doesn't necessarily mean anything awful. It just means this is something different from what we would expect. And so, maybe it means that you need to have specific follow-up care, or you need to go in for a clinical scan, something, or a different kind of imaging or whatever. But I think this surprises a lot of people that you would see it at this high of a rate in, again, otherwise very happy, healthy term kids. In our studies, we scan them a little later. Our rate, I think right now, is about 10% of incidental findings. And the vast majority of those are, we've had a couple Chiari malformations, and I'm trying to think of the other ear infections. That's always a fun one, because if it's an ear infection, that means we scan them. And of course, we used hearing protection to put pressure on their little ears, and it always makes me cringe that we did that when they had ears full of fluid. It just hurts. trying to think of some of the other things that we've seen here. I think it's really primarily Chiari malformations. We've had a couple of pituitary masses, but we don't see these kinds of things in general. Some of those kind of punctate white matter lesions, we'll see a few of those, but not a ton. And what I wanted to talk to you all about is kind of what to do if you have a patient that comes in and says, hey, I was in a research study, I got notification that they saw something, to kind of give you an idea of what we expect as researchers or what we hope happens. And then, I'm happy to answer questions or honestly take suggestions. That's even better. Because right now, when we get an incidental finding, again, from the radiologist, I will reach out to the family, I'll call them, they get a letter that describes what the radiologist saw. We, of course, have to put the caveat that the data were collected for research purposes, not for any kind of clinical identification. And we also give them the actual report, and then we say, talk to your provider and see what next steps are. Part of that is because we are not part of an academic health center, right? So Virginia Tech is completely separate from Carilion, and so we don't have those direct lines. Ideally, every project would have a pediatric provider that I could reach out to when we have an incidental finding and say, hey, and usually the radiologists are really good about this too, is this something that I need to get on the phone with right now? And do they need to be going somewhere immediately? How urgent is this? And so actually for one of the larger studies that we have ongoing, the Healthy Brain and Child Development study, we do have a site pediatrician. So Cameron Johnson is serving in that role for us. And it's been really, really helpful that I can contact her because she's on all of our IRB paperwork, so she's allowed to see names, she's allowed to talk to me about all of the findings. But if you don't have somebody in that role, it can be really disorienting, especially when you're trying to talk to parents and I can't say anything. I'm not a neurologist at all. I can tell them the image quality is great, all of the parts that should be there are there. But of course, in the case of something like a pituitary mass, I don't pick up on those things. So I'm not trained to, right? I haven't looked at thousands of images. And so when I am talking to families, it's really, I think it's unfulfilling for them because I can't answer their questions. And then of course, it's going to be really difficult for them to get in touch with you all immediately. So again, ideally you have somebody on your team that you can talk to and then they can connect directly with families. So I'm not in the middle as this weird, not helpful kind of person, because we all want to be helpful, right? So if a patient comes to you with an incidental finding, I encourage you to reach out to the researcher. So we can tell you, how did the scan go? Or, we oftentimes have behavioral measures as well. So for us, one of the most common behavioral measures that we get is the Bailey. And you all, I'm sure, are familiar with the Bailey. And so we have all of that information. And of course, that's standardized. You have to be certified to administer it. And so that may be helpful context. So I always encourage physicians, don't hesitate, and I always put my cell phone number on all of those letters so that families can call me, but you all are welcome to call as well so that we can have a conversation. The important part there is that we have to have a release in place so that I'm allowed to tell you that they were part of the study. And that's usually we get that at the beginning of a study anyway to facilitate that kind of conversation. But we also will remind them that we won't say anything about medical anything to even their physician unless they've explicitly told us we can. So we're very, very careful about that. But we also want to make sure it's not a barrier to care. So we try to proactively collect those kinds of releases. But keep in mind that the images were not designed for any kind of clinical anything. I think in general, they're typically higher quality because we just have more time, right? So our T1s and T2s are each 4 minutes long, and we're at 0.8 millimeter isotropic resolutions. That means every voxel or 3D pixel is 0.8 millimeters on each side. That's really high resolution, beautiful data. So I think it's complementary, but certainly we're not using contrast, right? So if you needed to see something specific, that would require another clinical scan. But we are happy to share images, again, as long as we have permission from the family, but please don't hesitate to talk to us. If you want to know what sequences were used, just any of the things, please call and ask. There's, and all of us, I will speak for all researchers, we don't want to stand in the way of care, especially for kids where it could be something that you can address early and well, right? We want to be helpful. So just call us, even if it's after hours, call us and let's talk about it. Because again, we want to make sure that you have what you need to continue their care. Okay. All right, so I'm going to talk a little bit about function. So it's all about structure because again, MRI is the best way without contrast or without ionizing radiation that we can look at structure. I'm going to try not to belabor this because I'm not, I like to joke, a functional person. I'm much more of a structural person. But the brain functions, and that function is important, right? So we're going to talk about these 4 methods a little bit, and I will walk through this really nice summary figure that one of my graduate students made. Because I think it's really helpful to contextualize this. Because again, at the end of the day, even if we're thinking clinically, we want to be able to capture something that's meaningful. And with functional data, that's much trickier than with the structural. Because with function, the brain is responding to something, right? And whether or not you're controlling what that something is, or maybe you just want to see what it's doing when it's, at rest, that depends on again, what we think is important. So the thing I will highlight is you notice the color on these boxes. So red is saying that it's not possible to get awake natural behavior. And for MRI, I mean, hopefully that's clear that most people don't lay in tubes awake to do things ever. That's just not natural, and we just don't do it. So it's not possible with MRI currently. There are, vertical systems and things like that, but even then, it's such an artificial environment that I would argue that that's not natural behavior. If we start to move into some of our other modalities, so things like EEG or MEG or FNIRS, we can have them be awake and behaving, but there are potentially trade-offs in resolution. So the y-axis here is spatial resolution, and the x-axis is the log of temporal resolution, so higher is worse. So fMRI, we have great spatial resolution, awful temporal resolution. Temporal resolution is really important depending on what you want to look at. So if you're interested in something like epilepsy, fMRI isn't going to help you much. Number one, there's motion, potentially, right? But also the way that the brain is functioning during a seizure happens really fast. And so we aren't going to be able to capture those patterns well with a method like fMRI. That's why usually EEG is used for clinical monitoring for epilepsy. The FNIRS is okay in both temporal and spatial resolution. You can only get down to the cortex though, you can't get deeper. And so depending again on what you're interested in, not going to be super helpful. And what I'm going to end with, again, towards the end, is talking about optically pumped magnetometer, MEG. That's a method that my lab is trying to develop applications for in infants. And you have a great balance there between spatial and temporal resolution. I'm going to go through these really quick. So functional MRI, this is the blood oxygenation level dependent signal that we're picking up on in fMRI. And so that's just based on the premise that when the brain is doing something, the region that is doing it will require more energy, and so in oxygen. And so we'll push more oxygen to those regions. And so we can measure that using fMRI because thankfully oxygenated hemoglobin has a different magnetic signature than deoxyhemoglobin. And so we leverage that to get an idea of what brain regions are active when. The picture on the left here is just showing that the network connectivity is in place pretty early. So this specific image is of the default mode network. So that's a network that we think is involved in basically kind of organizing the brain's function. It's trying to make sure that all of the other networks are working the way that they would anticipate them working, and that is in place in infancy. There are some places where infants are more active than adults and adults are more active than infants, but in general, it's pretty remarkable that that structure exists so early, and they've shown this in neonates as well, and so that's kind of exciting, right? The other side here is just showing what is more typically used in the clinic, if at all, and this is task-based. So this is where you're controlling the kind of stimulation that you're doing. So it could be listening to words. So language lateralization is something that's often done clinically, especially pre-surgically, so you can identify what parts of the brain in that specific person are responsible for language or motion, so or motor behavior. So eloquent cortex is what we call it. So you can use fMRI for that, but again, there are lots of limitations because it's slow, it requires an MRI. Again, you may have to sedate them. You can do auditory tasks in sedated kids and infants, but the signal is lower. So it's not ideal at all. And so to get around some of that, I'm going to skip that time. So to get around that, we can use something like EEG. And so the other thing I didn't say about fMRI is that it's an indirect measure of brain activity, whereas EEG is direct, right? So EEG is measuring the electrical The electrical impulses that are generated by groups of neurons firing together. Again, that's a direct measure of neuronal activation. Whereas the gold signal, again, is this kind of multi-layered, you have all of this concerted firing, and that requires more oxygen, and that's what we're measuring. So there are differences in vascularization, in blood pressure, in blood volume. All of those things can impact your gold signal. They will not impact, however, the electrical signals themselves. EEG, especially in babies, is kind of plagued by this issue of the signal itself, though, so that electrical signal is impacted by the kinds of tissues that it has to move through to be sensed. And so that's why in like epilepsy monitoring units, they'll put electrodes down because we want to know exactly what are the electric patterns happening inside the brain. If you did surface recordings, number one, you aren't sure where they're coming from. And that's, of course, very important when you're talking about an epilepsy and potential resection, right? But second, you also don't know necessarily what it means because the signal is, again, degraded. That's especially hard for babies because of the fontanelles. And so there's even another thing that we really can't model particularly well. I put here the side just in case you're interested in the brain waves that you can get from EEG. And of course, a gratuitous picture of an adorable baby wearing an EEG net. So for that big project I talked about, we're also getting EEG measures. And then this is one example of a naturalistic study that you can do with EEG, and they're just looking at pacifier sucking, which I thought was kind of fun. When you start to think about what kinds of behaviors would you think you might see differences in, it probably isn't going to be something super cognitive in an infant, because infant cognition is very hard to measure. But let's pick up on differences in sucking versus not. And so all they're showing here is that they did see a predictable change in the brain waves present at these two specific regions marked in red here during sucking and not sucking. So it might just be a nice task. It's also just a proof of principle that you can use EEG to look at naturalistic behavior. FNIRS is an optical method. Optical methods are really, really attractive because they're cheap, relatively speaking, right? They're portable. You don't need special shielded rooms or anything. All you need is something to produce the light and then something to pick up on the light when it comes out of the head. So the idea here is you're still measuring blood flow, or not really flow directly, but you're measuring the absorption of that light by oxygenated hemoglobin. And then you're trying to see how does that change in the areas where I expect to see change either during task or, again, during rest. You can combine these with some other optical methods to get things like cerebral blood flow, which is really important, especially in places like the NICU. And this is often used in the NICU in combination with some other technologies. But what I wanted to highlight here is that it's also applicable in places where you don't necessarily have MRI scanners or access to lots of technology. So this citation is looking in lower to mid-income countries and applying this technology, which is really exciting. You can combine it with them awake, and you can do a bunch of task stuff. So again, I think it's important to think about all of the different methods that you have access to, and depending on, again, what your questions are. But the main issue here is that, again, you're limited to cortex. You can't go any deeper than that. And there are some systems that claim that they can go deeper than cortex. I don't really believe them, at least not reliably, right? Because you still have to model what tissue that light is going through, and that's so variable from child to child, and it can even be different depending on if they're sitting up versus lying down. And so it's just hard to model the signals here. I did want to highlight, though, that you can do hyperscanning. And so hyperscanning is whenever you have two people and you're scanning them at the same time and seeing how their brains are responding to each other or to shared stimulation. So here's just a reading task with a mom and her two babies. And again, that's one of my research interests is synchrony. So the last modality we're going to talk about is SquidMed. So these are what does the S stand for? I can never remember. Superconducting quantum interference device. Don't worry too much about it, but just know that these are sensors that need to be cooled with cryogens, just like an MRI does, right? So it makes them expensive, it makes them bulky, and it makes them hard to do anything naturalistic. So this panel on the right in the citation is a really great description of what MEG is used for clinically for pediatrics. The issue here is that the signal that you can pick up with MEG sensors although it's not degraded based on the tissue that it goes through. So that's great, right? So that gives us one up from EEG. And it is a direct measure of function because, again, I don't know if you all took physics or remember back to the days of physics. Whenever you have any kind of current, based on the right-hand rule, you also get a magnetic field. And so all MEG is doing is instead of measuring the electricity itself, it's measuring the magnetic field that's produced. And so that is, again, the benefit there is that it's just a function of the distance from the source to the sensor. So if we can get sensors really close to the source, then we're good to go. And we have high temporal resolution and high spatial resolution. SQUID MEGs are difficult because the doer is, that's that big piece at the top where you would put your head. It's a set shape, right? And so you can either have an adult system that's way too big for a baby or a toddler, or you can have a child system, the pediatric systems that are over here, but then you can only use them up to a certain head size. And so, they're not often used because they're expensive, right? So some of the bigger children's hospitals, so Bill Gates, not that Gates, but Bill Gates, he's at Children's Hospital Philadelphia. And so they have a bunch of, I think they have 2 SQUID megs. But the idea there is that, again, we can get functional data that's meaningful, interpretable, and this is probably the most often, if they have access to a system, the kind of modality that's used for things like identifying eloquent cortex before pediatric surgery or brain surgery. And then on the left, I just always like to highlight hyperscanning because it's really interesting and fun. And so you can do that with a MEG, but of course you have to have 2, at least if you're using SQUID, and you have to, in here they're not interacting directly, they're just looking at pictures of each other. But let's see that that's also possible. Okay, so kind of to summarize some of the clinically relevant ways to measure brain function. So again, most of the time it's not used a lot, and that's mostly because it's very, at least based on the empirical evidence, there are very few instances where we know how to interpret that brain function signal, right? So something like epilepsy, that we've got, right? We know what an epileptic seizure looks like at the level of the brain. Sleep, also, we know what that looks like, right? We can identify different sleep stages. And then again, for surgical planning, depending on how old they are and what you're looking at. But when we start to think about things like just high-risk populations, so kids that are high risk for CP, we don't know if we got an fMRI of all kids that are at high risk for CP, we don't yet have a neural signature that tells us anything important, right? It doesn't tell us, are they at higher risk for hemiplegia? Are they at high risk for developmental delay? We don't know, and that's partly because the studies haven't been done, but that's also partly because of how messy functional data are in general. And so again, on the back end, people are trying to figure out how can we disentangle the signals to know where they're coming from, and then on the for researchers, we're trying to figure out, are there tasks that we can run that are meaningful for this versus just kind of seeing what the brain is doing. And sometimes that resting state information is really important, but it's just really hard to interpret sometimes. So really what we need is a modality that allows for motion and naturalistic behavior. So again, something we can just put on, get the information that we need, and then at least for research, understand mechanistically what's happening, and then clinically, hopefully translate that understanding to something where you can get a non-invasive measure and be able to treat your patients better. That's the goal. And so I'm going to make the case that optically pumped magnetometer magnetoencephalography is that modality, specifically for infants. Again, that's the space that we live in. I'm really proud to say that we're the only group in the world that is using this technology in infants of this age. So there are a handful of others, literally 4 or 5, that are looking at neonates, and that's again because they're pretty easy. You feed them and you lay them down and they're pretty happy. If you go, so we're looking at kids from 3 to 9 months. They're a little harder to keep engaged, right? You all have worked with babies. Just to give you a sense of what the system looks like. So again, we're measuring the little tiny magnetic fields using these sensors that can operate at room temperature. And that's the real innovation here. Because before, like I said, those SQUID systems have to be cooled with liquid helium typically. So that makes them expensive. And again, you can't move them. It's not portable for a reason that I'll talk about in just a second, but they are wearable. And so they can move around, which is pretty exciting. The other part that's exciting here is that we can get the sensors really close to the head. And so that means that our signal is going to be the highest that we can collect outside of the head, which is important when we're trying to figure out where the signals are coming from. But the first hurdle that we had to overcome was that there is no hardware to do this. And we had sensors and we had babies. You can't just like screw it on. Although some of the first measures that people got, they literally did just tape sensors on to the babies' heads, which is not desirable. It takes a long time to get them on. They don't stay. And so we had to figure out how do we get sensors in a place, because we need to know where they are and their orientations, or we can't model where the signals are coming from. So my team actually developed this headwear, and it's patent pending, which is really exciting as an academic, because we don't often get to, at least I don't get to do that. Engineers get to do that constantly. But we were really excited because it was a solution that worked for us. And the ideal situation is that we'll be able to produce these to scale, because they're also much cheaper. So each sensor, the new generations are about $12,000 per sensor, which again sounds like a lot, but not when you're talking about comparing it to an MRI system, something like that. Although again, we still can't do structure. All right. The magnetic fields that we're measuring are really, 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 really tiny. So you have to make kind of a hole in the Earth's magnetic field and with all the other magnetic noise that's happening. And so you have to have what's called a magnetically shielded room, or we have specifically a mu room because it's shielded with mu metal. And that's really the biggest cost here, that you have to have some sort of shielding. Otherwise, you won't be able to measure the teeny tiny magnetic fields that we're picking up. There are some folks, some physicists working on doing ambient field measures, but those are very, very, very far away from being applicable to anything. I think they've done some field testing literally in the middle of nowhere fields, because you're trying to get away from all of the electric noise, and you have to be at a very specific orientation to the Earth's field, and it's not there yet. They're working on it. Okay, I'm going to skip that because I don't think it's important. And we'll jump to just a little bit of some of our first data. And again, these are data that nobody else has ever collected, because babies move, and that does stuff to the signal that we're able now to model. And so just to show you kind of the two situations we're using. So the top is my graduate student, Megan Evans' dissertation project. So she's interested in kids that are at high risk for neuromotor impairment. And what she's doing is doing the GMA while they're hooked up to the system. And so hopefully, again, she's going to try to answer that question of, are there patterns that we can pick up on using this non-invasive technology that will predict later outcomes? So hopefully providing clinically relevant information to guide care, right? And then on the bottom, this little dude is just wearing our regular cap, but he looks very surprised. We're just doing some basic sensory tasks to validate the system. And over here, I don't know if anybody's used to looking at power spectrum density plots, but this is very normal. This is really exciting because it means we're getting data that are potentially robust, right? So this literally, can we get something that makes sense from having these sensors on the baby's head? You'll see the peak at 60 hertz. That's just the electricity going through the room. So we usually will filter that out. All right, and then I want to share some of our hyperscanning data. And so this is a really super simple task where we have baby on mom's lap, and she is looking at a screen, and she's told when she sees a crosshair to squeeze baby's hand. And so the idea here is that we would get a very predictable signal from mom, because it's just a very simple visual cued motor task. But then we would also get the sensory motor component from baby, because baby is feeling mom squeezing. And so what we're able to see is that we get actually noisier data from mom, which is kind of unexpected, but that's okay. But that if you look at panel C, that this is mom's source localized data, that is somatosensory cortex. So again, we're picking up meaningful signal on mom while she has a cap on, baby has a cap on. And then this top panel B, this is a time frequency plot of the baby's data. And so the important part here is the move, that's when mom is squeezing. And then the rest is after. And what you see is this increase, so if you notice the yellow, so that's a % change in that specific frequency band. You see this broad band change, which is an alpha and beta, and those are somatosensory and attentional signals. And so baby we're getting interpretable data or signal from this sensory motor task using this new technology, which is really exciting. And again, the mom and baby traces kind of follow each other. So we're really excited. The last actual slide, so take homes. So again, MRI, I'm totally biased, but I do think MRI is a really useful tool, especially as we start to develop ways to deal with things like motion. that we have methods that you can assess brain function, but there's still a lot of work to be done to be able to apply those scans in a meaningful clinical way. People are working really hard on this because if we can have a non-invasive way of predicting what care a child will respond best to, that will be really, really powerful. And that's one of the things that as a translational researcher, we're really trying to help with. Then again, my group is working on developing OPM-MEG for use in pediatric populations, specifically focused on infants. And then the last point is, if you don't remember anything else, please remember that researchers love to work with clinicians. And maybe I don't speak for all researchers, and I shouldn't. I like working with physicians because it's really important to me that the questions that we're asking are going to answer things that are important clinically, right? We all, at the end of the day, again, want happy, healthy kids. And yes, science for science sake has a place, and other people focus on that, but my lab is very translational. We really want to make sure that what we're doing has, even if it's not immediate, right? Maybe if it's not, posthumously, that's fine. I can handle waiting. It will benefit people. That's the goal. And with that, I think we have like half a minute for questions because we have to evacuate for 8:30.
So I'm not sure if I missed it, but of those different modalities that you introduced us to, do you see any of those having the detail of functionality imaging that is comparable to the structural imaging we have now? And of course, we want that integrated.
So do you mean to answer questions about structure? So is it a proxy? So will we ever be able to move to EEG or OPM-MEG to take the place of structure? Is that?
We want both.
Both?
As clinicians, we want both.
So,
structure, exquisitely, we want to know how that structure changes with development and see the functionality, how that changes and influences with interventions.
Yes. So for that, yes. I do think that if we have both, then you'll be able to better link that structure with function. Whether or not that those links are not rigorous, but consistent enough to be consistently used clinically, that part I'm not sure. And the only reason that I'm not sure is because that functional piece is so dependent on context and even recent history. So again, I'm used to working with babies, and one of the hardest things that we deal with is teething. And it ruins everything because, there's spit everywhere, it hurts, people are giving them, of course, meds so that they aren't, they can have fevers, right? My kids have terrible diarrhea and diaper rash. And so when you're looking at an infant or any individual, right? So this even goes up to, adults that have disabilities where you can't necessarily ask them questions and things like that. You aren't sure what the brain's responding to, is my point. And so I think that if we can find out ways to standardize that context, and usually that's through tasks, right? So if we can design tasks that are very specific, then yes, I think that function will help us understand structure-function relationships in a meaningful way. So that's the issue here, is that there's a lot of what we call noise just because we aren't interested in it, and I really think it's just signal that we don't understand. And so if we can understand those signals, then I think we'll be much, much better off. That's why I like working with babies, because they're simple, relatively simple. They like eating, sleeping, being touched, and we can leverage all of that. That's why I like that pacifier sucking task, because it's a task, but it's just something that they do. And I think taking the brain out of context and asking it to do things that it's not really, at a developmental stage to do, something that's not salient for it, that's when we start to have results that we don't know what to do with. Thank you. Awesome.